Bom dia, pessoal. O Centro de Estudos hoje será mais uma vez no modelo híbrido, ou seja, presencial no auditório do Pavilhão Leão Tangini e virtual pelo Zoom e transmitido pelo YouTube. A palestra é parte do curso de LymphoRioMove 2025, que é um curso internacional de imunoterapias e ferramentas de laboratório na era da medicina de precisão, que é coordenada pelo professor pesquisador Vinícius Cotta-de-Almeida. O Vinícius é graduado em Farmácia e Bioquímica pela Universidade Federal de Juiz de Fora, tem mestrado em Biologia Parasitária e doutorado em Biologia Celular e Molecular pelo INSERM, Fiocruz. E pós-doutorado no Massachusetts General Hospital da Harvard Medical School. Vinícius é pesquisador titular do IOC, Fiocruz, cientista do nosso estado, bolsista da FAPERJ, bolsista de produtividade do CNPq e coordenador da equipe de imunologia médica do curso de medicina e do MEL, da Estácio de Sá, aqui no Rio. É também coordenador de Educação Internacional, vice-presidência de Educação da Fiocruz, coordenador do projeto e coordenador adjunto do programa CAPS 30 da Fiocruz. Ele desenvolve projetos científicos e de desenvolvimento tecnológico na área de imunologia celular, com ênfase no desenvolvimento e migração de linfócitos T, erros inatos da imunidade, bioimunologia e anticorpos terapêuticos. Como a gente pode ver, o Vinícius é um pesquisador jovem, mas já bastante produtivo e bastante ativo, né? Ele convidou para apresentar a palestra o Dr. Renaud Lesourne, do Instituto de Doenças Infecciosas e Inflamatórias de Toulouse, na França, que vai apresentar a palestra Sinalização de pontos de verificação imunológica e mecanismos de regulação. E também o pesquisador Dr. Nakaya, do Hospital Israelita Albert Einstein, do Brasil, que falará sobre IA e medicina de precisão. A mediação será feita pela nossa colega aqui do IOC, do Laboratório de Timo, Adriana Bonomo. Queremos agradecer ao Vinícius por trazer ao Centro de Estudos e também um tema tão importante, tão na moda, né, digamos assim. E também aos palestrantes, o Dr. Renaud Lesourne e o Elder Nakaya. As pessoas que estiverem assistindo online poderão fazer perguntas pelo chat, que serão encaminhadas aos palestrantes. Então, boa palestra, boa sessão a todos. Vinícius, pode dar continuidade, por favor.
Muito obrigado, Marli. Thank you very much. Okay. Good morning, everyone. As you know, this is the equivalent seminar series of our institute. And I really thank Marli, that is the organizer of this seminar series of our institute, for getting us here today, because I think it makes a lot of sense, not only for the students, we have all the 21 students that are part of this course here, and of course the whole community, scientific community, can profit to have all this discussion with both of them, Renaud Lesourne and Elder Nakaya, that you know, are the professors today with you, that you can, of course, make your questions. And Marli already said, both are going to talk one after the other already. And after that, Adriana Bonomo and Zilton Vasconcelos is going to be the moderators for the discussions, for the questions in the chat. Thank you very much. Well, just a very quick introduction. I mean, Renaud Lesourne is already the second time with us here. Last year, he was also a professor. We invited him again to be here. He has been working at the Institut Pasteur and over NIH, essentially with immunoregulation, mainly working on regulation of T-cell signaling. And today he's going to talk to us about the immune checkpoint regulation. Thanks a lot. The floor is yours.
Thank you. So, bon dia, everybody. So my name is Renaud Lesourne, as Vinny said. I'm coming from the Toulouse Institute for Infectious and Inflammatory Disease. So just so you can know, Toulouse is in the southwest of France, next to the Pyrenees. And in the lab, we mostly interested by, these days, for the past 5 years, by all those receptors that are co-expressed with the T-cell receptor at the surface of T cells. and we try to understand on the molecular point of view how these receptors operate. And you see we're so interested by that we have the tendency to try to identify to those receptors and mimic how we project their structure can be. So sometimes you have very basic receptor, you can see the diversity of their shape, and sometimes you have more weird, represented by Loic here, structure for these receptors. So they're very diversified, and it's one of the things that we try to understand in the lab. So today I'm not going to really talk to you most about our recent results. It's more would be a lecture format. And I would like to give you a grasp of different aspects of what the research on immune checkpoints that have really revolutionized the therapy, particularly in cancer these days. I will quickly talk about how those receptors can be used in a therapeutic perspective. And as I say, I'm more interested into the mechanism in the lab. It's more the focus. So I will talk to you about how these receptors work on a molecular perspective. And I will try after to discuss about what are the unsolved problems, a new direction in the field of fundamental immune checkpoint study. And then I will finish by talking quickly about how those receptors that are regulators of immune system are themselves submitted to regulatory mechanism. So you know that one of the challenge of the immune system is to face and to direct immune response against a wide variety of different pathogens. And sometimes those pathogens can be very similar to the self, you know, and we need to find a way to discriminate between those potentially harmful antigenic structure and those that belong to us. So one of the goals of the immune system, you know, is to recognize those different shapes, recognize the self, but preserve it and not act against it and destroy the potentially harmful pathogen. So these are the two main challenges of T cells, and to respond to the diversity of pathogens and to be very specific in its response. And part of this is governed by this amazing receptor, the T cell antigen receptor, which you know can recognize an amazing variability of antigen. And one of the things that is important to keep in mind for students is that it does recognize self-antigen permanently, but it doesn't really react to those antigens. But it's important that it recognizes it to be able to scan cells from the organism. And when it recognizes like foreign antigen, this recognition triggers a signal that leads to T cell activation. So the T cell receptor, you know, is basically when it recognizes an antigen with sufficient affinity, it engages this kinase LCK that will phosphorylate the subunit of the T cell receptor and trigger pathways that are dependent on kinases and adapter protein. And those pathways, and what is fascinating with this receptor is that it can induce a wide variety of response, going from cytokine release, cytotoxicity, controlling T cell development, the migration, the differentiation of T cell, leading to exhaustion in some chronic diseases. So the TCR is really endowed with a great power in T cell, and you know the famous sentence, with great power comes great responsibility. So in fact, the TCR needs to be controlled by some mechanism. And the first layer of control is to add to the TCR a battery of co-stimulatory receptor that will amplify the signals from the T cell receptor, and that are also important to induce some more specific response depending on the context in which those receptors are engaged. So the T cells also, despite those first layer of control, can lead, you know, when it's not properly controlled, or in certain cases to autoimmunity, or excessive inflammation and tissue damage. And one of the typical aspects of immunity, I think, in general, is that you have all this layer of regulation that are engaged. And T cells are permanently submitted to those negative regulatory mechanisms that control and prevent the development of autoimmunity, for instance, or excessive inflammation. So there's many different regulatory mechanisms, but one of them is supported by those receptors, which are inhibitory receptors that are expressed together with the TCR on the T cell surface. We can name some of them that you probably all know: the PD-1, CTLA-4, LAG-3, TIM-3, TIGIT, that is not here, or BTLA. So they all, we'll see that they act by different way, but the way people represent it is that these receptors are basically inducing signal that will inhibit the signal triggered by the TCR or the co-stimulatory receptor. In fact, it's more complicated than that. And this is enhancing the threshold of T cell self-reactivity. It can reduce the magnitude of inflammatory response and also lead to T cell exhaustion. So these receptors are pretty famous, as you know, because it has been a revolution in the field of cancer therapy to develop the idea of developing antibody that would block this inhibitory receptor, because in the tumor environment you get this accumulation of T cell with an exhausted phenotype, and part of this effect is due to this accumulation of inhibitory receptor at the surface of T cells. So the idea of those very seminal therapy is to use monoclonal antibody to block the capacity of this receptor to engage their ligand and to reinvigorate T cell response in the tumor environment. And this lead to the Nobel Prize of Tatsuko Honjo and Jimmy Allison. So just a few slides on how those receptors, since this initial discovery, are now used as therapeutic levers in multiple clinical settings. You see this is a table that represents, I think it's from a paper, a review published like 2 or 3 years ago, and it has advanced since then. But you see how many of these receptors are now targets in the field of cancer with antagonist antibodies. Some are commonly used, like CTLA-4, PD-1, or LAG-3. And additional strategy, aiming and stimulating, this time not blocking, but co-stimulatory receptor are also very strongly developed. And now, one of the important clinical investigation is to see whether we can combine those different therapeutic antibody to promote even better T cell response and have a more targeted T cell response that can lead to less immune-related adverse event. You know that these receptor CAR T cell therapy is also a very promising approach of very more than promising in terms of liquid tumor, but also people are trying to improve how those cells work in the case in the context of solid tumor, and a lot of those CAR T cells express this variety of immune checkpoint and inhibitory receptor, and some recent papers have shown that when you shut down by genetic approaches those inhibitory receptors like CTLA-4 here or BTLA, you boost the capacity of CAR T cell to eliminate tumor. One of the hallmarks of those receptors is when you have a knockout for those molecules, you generally develop autoimmunity, exacerbated autoimmunity, either spontaneously or in more selective model of like multiple sclerosis or lupus. And of course, from that, people had the idea to, instead of blocking those receptors, to use agonist antibodies that would engage those receptors to stimulate their inhibitory response. And these were 2 papers, one in New England Journal, I think 2 or 3 years ago, where they used this agonist antibody against PD-1 to decrease the development or the pathology of rheumatoid arthritis in patients. And this is another paper where they generate, this B-specific LAG-3 antibody that can recognize LAG-3 and cross-link it to the MHC molecule, which is one of the ligand of LAG-3, to decrease the development of autoimmunity in mouse model. So this is also one way these antibody and these receptor are used. Finally, I couldn't talk here without putting a slide like that. It's just to show that those receptors are also very strongly involved in the regulation of T-cell response in the case of infection, HBV, HCV, HIV, also tuberculosis, malaria. So I don't think there's there start to be some research to see how immune checkpoint blockade in those different pathology can be helping to eliminate the virus, particularly in the case of latent infection like HIV, but I don't think these are really true clinical approach that are engaged, at least to what I know. So I just want now to describe you, what is more my field of expertise, is how those receptors operate. There's basically four molecular operating modes for those receptors. And the first one is the one that is always described. It's what I was mentioning a bit earlier, is this capacity of those receptors. This is the example of PD-1 to negatively regulate through signaling the signals triggered by other receptors. So you see those receptors are phosphorylated on specific inhibitory motif, and then it recruits those phosphatases molecule that are going to counteract somehow the pathway that are triggered either by the TCR or by co-stimulatory receptor, and PD-1 is known to be a very quite selective negative regulator of CD28 more than the TCR, which is very interesting because it shows that they're not just shutting down the T cell or the capacity of T cell to respond to antigen, but having a more refined effect on how T cell would respond to antigen. Another example here for TIGIT, you know that is also having motif, not exactly the same as PD-1, and that would recruit SHP2 and block and inhibit pathway that are triggered by these other co-stimulatory receptors, CD226. So the second mode of action is the capacity of this receptor, and this is changing the view a little bit of how these receptors are generally represent of interfering with the co-stimulatory molecule, co-stimulatory ligands that are expressed on cells that are recognized by T cells. The typical example is CTLA-4. So CTLA-4 is interacting very strongly with B7-1 and B7-2, which are the ligand of CD28 of this co-stimulatory receptor represented in green here. And it takes out by a process called transendocytosis those B7-1 molecule to degrade it inside cells. So that the APC, the cells that present antigen, is lacking those co-stimulatory ligands for CD28 and deprive T cells from being able to engage those ligands. This is not only the case of CTLA-4, you can see that with TIGIT, and you will see that mainly most of those receptors operate through distinct mechanisms to block T cell activation. And TIGIT, that I mentioned, was able to negatively regulate some signal by CD226, is also able to take and to, because it has a very strong affinity to the PVR, which is the ligand of CD226, and to capture the ligand of CD226, preventing CD226 co-stimulatory signal to be engaged to the PVR. So taking out, again, the ligand. A third mode of action is the capacity of this receptor to stimulate some immunomodulatory pathways. And this is the case, for instance, of PD-1. So PD-1 can initiate signal through its ligand, PD-L1, expressed on cancer cells. And this interaction with PD-L1 will trigger signal that will inhibit the capacity of interferon, for instance, to kill or to arrest a tumor growth. So you see that it's not the receptor by itself that is doing that, it's the capacity of the PD-1 to trigger signal through its ligand. Another example, for instance, comes from this study on LAG3. LAG3 is very strongly expressed on regulatory T cells, and it can promote the suppressive function of regulatory T cell by shutting down the expression of MYC, which somehow is something that is repressing the normal suppressing function of T cells. So you see here the receptor is not negatively regulating, as we would say, signal by the TCR, but diminishing the activity of Treg cells, increasing the activity of Treg cells. And the last mode of action of those receptors are the capacity to perform some kind of physical disruption of a stimulatory receptor complex, not acting in a biochemical way, but a more physical way. So another example of this is, for instance, LAG3 again. You see that LAG3 in fact contain an acidic region that acidify the environment around the CD4 and CD8 co-receptor that are recruiting this kinase LCK. And by acidifying this region, it prevents disrupt the interaction between LCK and CD4 and CD8 that relied on a electrophile amino acid. Another example, again, you see for TIGIT that has been shown in this paper is that you see that TIGIT is able to somehow prevent the dimerization of CD226, which is required to induce optimal signal after engagement of its ligand. So you see that one of the fascinating thing, and I think that's one of the unsolved questions that I'll put later, is that these receptors are described to operate through different mode, even for one single receptor. And I don't think we still know, what mode are the most important, and maybe they are coexisting and engage in different contexts. One thing that I wanted to mention, because I think it's important for the therapeutic point of view, is that these receptors do not necessarily need to engage a ligand to work. And they have a capacity to, just by being expressed, to trigger a signal that will regulate the T cell activation. And this was shown in a very nice paper from Christopher Garcia in Nature, where in fact he showed that PD-1 is just by being expressed at the surface, it's phosphorylated by the kinase, and the engagement of the ligand is able to somehow to phosphorylate the second motif of PD-1 intracytoplasmic tail. And what is interesting is when he does a knockout of PD-1 and not engage the receptor, just do a knockout and stimulate the TCR, you see that just by taking out PD-1, you have a higher level of response. And when you do the same thing but adding nivolumab, an antibody that block PD-1 interaction, you still see exactly the same type of response that is increased in the absence of PD-1. So in fact, it suggests that the use of this antibody blockade is good to prevent the interaction of PD-1 with its ligand, but does not abrogate all the inhibitory capacity of this receptor. And one possible reason to that is because the SHP2 molecule is able to bind in trans to two PD-1 molecule like that, even if this motif is not phosphorylated. So they made a very interesting tool where what they did is that they generate those B-specific antibody that can on one side block the interaction of PD-1 with its ligand, and on the other side bring in the complex the CD45 phosphatases that will dephosphorylate PD-1. And they did some therapeutic model where they compare, so this antibody, they call it the Reaper PD-1, and they compare it to the chemotherapy with anti-PD-1. And you see that they have a better effect, at least in this model. It was not tested in patients, but as compared to the basic anti-PD-1 therapy. So now I would like to do a quick overview of where I think research is going on a fundamental perspective on those receptors and what are the unsolved problems and new directions. One of the first things that is probably moving quickly these days is to understand what are these receptors doing beyond T-cell exhaustion. You know that they were characterized mostly in exhausted T-cells. They were very strongly associated with that. But now it's becoming clear that they have a more complex function than just inducing exhaustion in T-cells. This is an example of a paper where they did some analyzed the role of PD-1 in the context of acute infection with virus, and they show that PD-1 is very important to induce a memory response in CD8 T cell, preventing, by preventing the exacerbate development of effector T cell, which is probably required to attenuate the development of effector response to promote an efficient memory. Another example of these, the function of those receptors beyond T cell exhaustion is a recent paper in Nature in Immunology where they show that PD-1 is very important for inducing and specifying the development of TRM cell formation in the skin. So again, showing the role of those receptors beyond exhaustion. A second aspect of research that I think are particularly developed is that most of the study on those receptors have been on T cells. And you see when you look at the expression profile of those molecules on immune cells, they have a way more, they express beyond T cell in many other immune cells. And really nothing much is known about the function of those receptors in those cells. So just an example of recent paper, there was this study by Stuart Tangier and Jean-Laurent Casanova who showed the role of PD-1 in human and mice. They identify patients that have an immunodeficiency in PD-1, and they show that this PD-1 is very important at the surface of B cell. They did some B cell specific PD-1 knockout mice, and they showed that it's very important for the development of memory B cell response. So again, an unexpected role of PD-1. Another example of that is a recent article in Nature showing that therapy based on the blocking TIGIT is not acting only on T cell, but is very important to act on myeloid cells, probably to promote and to target and to lead to better anti-tumor T cell response. A third, I think, unsolved problem, a new direction, is that these receptors seems to operate in a very coordinated fashion. It's what can be seen in the context of exhaustion, where you have many receptors that are co-expressed on the surface of T cells. And more studies are trying to understand the interplay between those receptors at the surface of T cell. And this was an article, I think, published by Dario Vignali, where he showed that the LAG3 and PD-1 at the surface of exhausted T cell, this was shown in patients, but also in mouse model, they have some kind of synergistic cooperation or additive cooperation, where when you mutate one of these receptors or mutate individually or combine the mutation, you get you get response that are exacerbated and that are also selectively induced in the context where both receptors are blocked. This was also shown for the example of TIGIT and PD-1 in this article by Ira Mellman, where they show that PD-1 and TIGIT act together somehow to block CD226 signaling. PD-1 probably, what he claimed in the paper, inhibit the signals induced by CD226, and TIGIT will act by disrupting the dimerization of CD226 and promoting a homodimer. So again, a collaboration of those receptors together to control T cell response. One of the last thing I wanted to mention here is that one of the very interesting observations is that many of those receptors, it's the case for LAG3 or TIM3, have multiple ligands. And they also have multiple signaling subunits. And there start to be some evidence that depending on the ligand that the receptor is engaged, the signals that are triggered are not the same. So there's the idea that they're not broadly turning down signal independently of the ligand, but there's maybe a very fine tuning of what these receptors will inhibit when T cells are stimulated. And a last thing I wanted to point that I think is emerging is to understand the difference between what has been published in mouse model and what is seen in clinical context for human. And this is a nice study of Enfui, where, basically he tried to understand he showed that there's a very strong difference between the PD-1 expressed in human and PD-1 expressed in mice. And he found that there's a sequence in the human form of PD-1 that is very stabilizing the interaction with SHP2, which enable this receptor to have a more stronger inhibitory capacity in human T cell than it does in mouse. And there's more and more papers showing that there's molecular differences between those receptors in mouse and human. So the last part of my talk will be on what I start to work on a little bit in the lab. So those receptors are regulatory receptors, but you may understand that they need themselves to be very strongly regulated, because if you have too much inhibitory pathway, T cells are not able to respond properly in multiple contexts. So in fact, these receptors are very tightly regulated themselves. And I just wanted to provide 2 examples of that, and then I will briefly mention some of results we had recently in the lab. This was a paper published in Cells a few months ago, which showed that, in fact, you see on LAG3, those receptors are somehow, some of them are shut down at steady state. And they show here that LAG3 adopt a close conformation at an intracellular level with a small sequence that make the intracellular the sequence domain of LAG3 interact with the membrane, preventing the access to the inhibitory motif of LAG3. And what they show is that, in fact, this intracytoplasmic domain is ubiquitinylated by this molecule, this ubiquitin ligase, called CBL. And this opens up, general ubiquitinylation leads to degradation, but here it doesn't do that. It opens up the tail of LAG3, and it enables the inhibitory function of LAG3. And they show very interestingly that there's a correlation between CBL expression, the ratio between CBL expression and LAG3 expression in the context of therapy, and that the more you have CBL and the less LAG3, the less patients are responsive to therapy aiming at blocking LAG3. Another famous example of that is for PD-1. You know that PD-1 is expressed extremely early after T-cell activation, yet when you do a PD-1 knockout mice, you don't really see an effect on priming. And this was explained by Okazaki here by the fact that, in fact, PD-1 is somehow repressed. The capacity of PD-L1 to interact with PD-1 is constantly repressed by CD80, which is this ligand of CD28. So when you have expression of CD80, it interacts with the ligand of PD-L1, and prevent the interaction of PD-1 with PD-L1. So the level of inhibition by PD-1 is going to be very dependent on the level of those ligands that are ligand for co-stimulatory molecule. And to conclude, I'm just going to end with an example of a study that we published in the lab last year. And so we have long studying this molecule called Themis, which is a very unusual molecule because it has domains that are called cabit domains. And those cabit domains bind, in fact, to the phosphatase domain of those tyrosine phosphatase that I mentioned before, SHP2 and SHP1. So Themis is very, very effective to block SHP1 in vitro and to block the catalytic activity of SHP1, but less effective to block SHP2. So one of the main characteristics of Themis is you have this drop when you do a knockout on T cell development. You have a decrease of CD4 and CD8 T cell and a very strong lymphopenia at the periphery. So for long, people have tried to understand what Themis was doing. And a student in the lab, a PhD student, Suzanne, investigate, in fact, we asked the question of, if Themis can block those phosphatase, can it block simply the receptors that are engaging those phosphatase? So those receptors are PD-1 that recruit predominantly SHIP2 and BTLA that recruit predominantly SHIP1. So to ask this question, we started by a very simple experiment where we cultivate T cells with an anti-TCR antibody with a molecule that are stimulating those receptors, here BTLA agonist antibody. And we compare at the proliferation in wild type or Themis knockout mice. And we showed here that when you are in wild type mice, you see when you add an agonist antibody against BTLA, you basically don't much block the proliferation or very little. When you do the same in a knockout mice for Themis, you see that you exacerbate the capacity of the receptor to inhibit T cell response. And this is particularly true in CD8 T cells where the receptor is somehow inactive in wild type mice and becomes able to inhibit when you take out Themis. So we tried to do the same kind of assay with PD-1 and we were hoping that it would work because it would be a fancy way to find a natural inhibitor of PD-1. But we noticed that when you compare the inhibitory function of PD-1 in a wild type, in a Themis knockout mice, or even in cells that are overexpressing Themis, PD-1 is still able to inhibit very well, and there's no major change in the capacity of PD-1 to inhibit T cells. So we try to understand how Themis could regulate the signaling function of BTLA. And what we showed is that Themis, in fact, is recruited directly to, maybe show the image, I think it's, Themis is recruited directly to BTLA through GRAB2, and it's co-recruited with SHP1 to the intracytoplasmic domain of BTLA. And then we try to understand how Themis do to block this receptor. So Themis, in fact, was shown to block SHIP1 by promoting its oxidation and promoting the oxidation of the catalytic cysteine. So we analyzed the oxidation of this catalytic cysteine in the wild type of Themis knockout mice. And what we did here, we immunoprecipitate BTLA and we look at the co-precipitation of SHIP1. You see that in the absence of Themis, you have fairly the same amount of SHIP1 that associate with BTLA, but you see that the level of oxidation of SHIP1 is decreased. So somehow revealing that the phosphatase has probably an exacerbated phosphatase activity because of this decreased oxidation of the catalytic cysteine. So Themis basically blocked the receptor by promoting the oxidation of this catalytic cysteine. So we now wondered whether we can find some more physiological relevance to that. And as I mentioned, Themis is very important for T cell development, and we wondered whether Themis was somehow promoting the positive selection of T cell, promoting TCR signal by blocking the inhibitory function of this receptor. You know, it's like in mathematics, minus per minus is a plus. So it's basically how Themis is working. So to address this question, we cross the Themis knockout mice with BTLA knockout mice. So we expect that if you have, when you take out Themis, you have exacerbated BTLA function. But if you take out BTLA in the context of Themis deficient mice, you should restore positive selection and T cell development. So that's what we did. You see, we have mice that are either deficient for Themis, or deficient for BTLA, or deficient for both Themis and BTLA. You notice this disruption of T cell development in the Themis knockout mice. You see that you have a strong lymphopenia at the periphery that is very characteristic. And when you delete BTLA in the Themis knockout mice, you restore completely the number of CD4 and CD8 T cell in the thymus and also at the periphery. So suggesting that Themis is in fact promoting T cell response by blocking this inhibitory receptor. So that's all for today. I just would like to show you the team that I'm co-heading with Loïc Dupré, that many of you know very well here. And I would like to thank Loïc particularly because he is the one who got me involved in and participate to this seminar. And also would like to thank very much Vinny for inviting me and giving me the opportunity to talk here. Thank you.
Thank you, Renaud. Thanks a lot. And right away, we are going to invite Elder Nakaya then, that you know already for some talks that he has been here at Fiocruz. He's a researcher, as Marli said in the beginning, he's a researcher at the Einstein in São Paulo and also at the University of São Paulo. And many of you know of as a big data scientist that we have in Brazil, integrating data and working these systems, immunology, and other very interesting projects. And most of all, he always liked to put a lot of insights in the students, in the researchers, and in all the community. Thanks a lot for coming.
Thank you.
And at the end, we need to sing her birthday, because today he came here even in the day of his birthday. We can do this already. Happy birthday to you. Happy birthday to you.
In French, please. I want to hear it in French. Thank you, guys.
Thanks a lot for coming, and take your time with your family to be here with us. Thanks a lot.
Birthday gift. I'm very pleased to be here. But thank you so much, guys. Good morning. I would like to show you a little bit of the stuff that we have been doing in our lab in AI. Of course, nobody is on the stage yet to change with nanorobots, but you are aware that there is a train coming, and this train is named artificial intelligence. And very soon, any scientist will have to learn a little bit of data science. There is no other way. You don't have to program, but you have to know data science. You will not need to know how to program, because programming language is a language like any other language. So the same way you can translate French to Portuguese, you can translate Portuguese to Python or R. So very soon, what you gonna witness, and I bet you that Vinicius is gonna become a hacker next year or perhaps this year. And one example of this is that we started a project last week, literally last week, and it was inspired on this problem. Imagine that there is a person, a boss, that wants to know how many children between 7 and 11 were hospitalized with dengue in Butantan last year. What the person has to do, call the IT person that is going to have to download a huge database with 21 million cases of dengue, and then it has to write a SQL query that's going to retrieve the number, and that's going to be like 17. And then it tells the person, the boss, that is 17. What we did last week was to remove this guy and put a AI there, which is basically the first thing we did was to transform that table into a JSON file, which is a structured way of you organize the table that's going to tell the AI how it has to handle these files. And now when the person asks, for example, how many dengue cases exist in Pará, it's going to generate the codes and make the graph for the person. So meaning that the person can talk whatever they want, and the AI is going to already give the results to the person. So that's why I'm saying that very, very soon also Zilton is going to be like using like a crystal ball to talk about genomes and rare disease. So in the past 10 years, what we have been doing was to do the other way, which is making user-friendly tools that allows anyone to do this kind of science. And I split this into 5 parts, but I'm going to focus on the precision medicine part, which is the first one. And this is inspired by the fact that if you have a group of people that has the same disease, let's say cancer, or is infected with the same pathogen, they may have completely different outcomes. Some are going to be fine, some are going to be sick, some are going to die. And that happens because every single person is unique. You have a different immune system, different environmental factors that influence your outcome. And if you measure the activity of millions of biological components, then you're going to have this problem, which is a hairball. And what we do in the lab is try to find what is the minimal part, the small part of this network that can predict the outcome. And we have been focused mostly on infectious disease and vaccines, which are relevant to Brazil. But we are also using this machine learning approach to SUS, to our universal healthcare system. But instead of expecting that we're going to sequence 100 million people, the genome of 100 million people, we can actually use the same algorithms to analyze the metrics, the lab exams, clinical data that is already collected by SUS. So we developed some machine learning tools that are novel, that nobody did, and we are also using state-of-the-art machine learning tools. For example, this one, Graph Neural Network, is the same tool that Netflix used to tell you which movie you're gonna like, and TabPFN is the equivalent of a ChatGPT for tables. Instead of training with words, you train with tables. And when we use the TabPFN in dengue cases, we can tell who is gonna die or be hospitalized by dengue, or at least try to predict this, by using only the symptoms, comorbidities, and demographic data. Meaning that before the person goes to see a professional, he already can estimate what is going to be the risk of dying of dengue. The other part of the lab that I don't talk often is the sexual dimorphism of disease. You know, and that impacts a lot the human health. And we start this by analyzing the genes between healthy and healthy men and healthy women, and look at the expression levels of the genes between these two genders, the two sexes. And then what we saw was that many genes were actually related to the immune system, and not the ones that are in the X and Y chromosomes. In Cesar, what he was focused on was on juvenile idiopathic arthritis, which is an inflammation of the joints, and it's under children under the age of 16, and it is idiopathic, meaning that you don't know the cause of the disease. And because you don't know the cause, there are over 400 studies of omics on juvenile idiopathic arthritis. And what we are trying to understand is why there is more women with juvenile idiopathic arthritis than men, and why it takes longer for women to respond to treatment compared to men. And what Cesar did was to pick 9 studies of juvenile idiopathic arthritis, and then we analyzed all of the studies in the same way, trying to find the common signatures that were upregulated in men or upregulated in females. And then we rank the genes that were more expressed on females on the right, or more expressed on the males on the left. And when we started looking at the genes that were highly specific to females, we saw neutrophil degranulation. That means nothing. Just suggests that the neutrophils are related. And then to confirm that neutrophils were involved, we look at single cell data, and then we look at specifically where these 44 genes were expressed in this single cell data. And then we found that they were highly expressed in neutrophils, confirming that this is truly neutrophils. The next question is, which neutrophils? Most people look at the mature neutrophils, but there are immature neutrophils that usually in inflammatory conditions go to the bloodstream. So we went to another paper where they sorted neutrophils from different stage and did the transcriptomics. And when we compared the 44 gene signatures, we found that actually they were immature neutrophils. And then we look at the differentiation process happening on the neutrophils differentiation. And we also use this as a score. So if you take that expression as a score, and then you measure the expression, for example, in male with no treatment or placebo, there's no change. Female already have a higher score than male, you see? And then when you treat with anakinra, which is an IL-1 beta inhibitor, the scores in males go negative in 1 month and even more in 6 months. But if you do the same with females, it doesn't change. Meaning that probably the delay in response to the treatment has to do with immature neutrophils. And then the next question is why? Why females have different amounts of immature neutrophils in males. And how can we test this? Well, we can get a transcriptomic data from women and men over before puberty, after puberty, and menopause, and then we see that this change. In another situation where you have very controlled amount of sexual hormone levels is pregnancy. So we took a transcriptomic data of women pregnant, and then we saw that during pregnancy the score increased, and after the baby is born, it goes back to normal, proving that sexual hormones is probably what is behind this. And then we published this paper with zero reais of reagents, because all of these were done with public available data asking different questions to different data sets. We also work a lot on COVID, and I'm gonna only talk about one study, which is Jonathan's study, where he took people that were transcriptomic data of severe COVID. And that was not novel. Actually, there were 56 studies of transcriptomics of COVID. But the problem is that if you take 2 studies at random, and then you look at the genes that were differentially expressed as reported by the authors, you're going to see that the overlap of them is minimum, meaning that anyone says that your genes are related to COVID. So what we did was to download everything, reanalyze everything the same way, and then we tried to create an atlas by looking at not only, for example, one study of blood of COVID, but actually 8 data sets of blood transcriptomics. And then we identified the signatures that were robustly changing in severe COVID in the blood. And then we can apply the same thing for the other tissues. And when we have the signatures that were consistent in different tissues, we can combine everything into this network. And then we have to show that these insights are useful for people. And then we pick one gene that is called RAB8B, because RAB8B is from a RAB family, which is a family of proteins that is important for the endocytosis and exocytosis of the virus. But nobody knew anything about RAB8B and SARS-CoV-2. So we established a collaborations that did knock out with CRISPR RAB8B, and when you infect, the replication goes down, meaning that this gene is important to SARS-CoV-2, and that analysis help us to find. We also create a dashboard. So if you go to this website here, or you can look at in Instagram, and then you can see, you can type the gene that you want, and that's gonna tell you if the gene is differentially expressed on a specific tissue. We have this project called Metatranscriptomics, which is basically was started with André, that is trying to do the microbiome, precision microbiome, and Icaro is trying to merge the host genome with the microbiome. So what it does is we get bulk RNA-seq data reads, and the first thing people do is to map to the human genome. And then the unmapped reads, they throw away. What we do is just to see if these reads actually map to the microbes. And now we are running in almost 60,000 human samples to find the microbes that are there. And we are also doing the same thing with single cell data. So this is a leishmaniasis paper that we submitted, where we generate single cell data of cutaneous leishmaniasis and look at what's going on there. But we also apply this single cell metatranscriptomics to find cells that were potentially infected with the parasites. We can also apply this to other diseases, for example, IBDs, and then we can see bacteria that were probably involved with the disease, and that is at single cell level. We also have new ongoing single cell projects. One is rheumatoid fever, the other one is dengue acute infection with type 2 diabetes, and the other one is cutaneous leishmaniasis using spatial transcriptomics. I don't know if Herbert's, oh yeah, he's helping me to do this. So oncology is of course very much related to precision medicine, and you all know the problem of the microenvironments, the tumor microenvironments. In how can we study this? We can get the tissue, and then we can use pathologists to look at the tissue using image, or we can try to use flow cytometry, in situ hybridization to look at the cells. But we can also do bulk transcriptomics, and recently we can now use single cell transcriptomics that merge these two technologies here. And more recently, the spatial transcriptomics that combines also the image parts. And one example is this paper here that was led by Carlos that was studying brain metastasis. What we wanted to know is why brain metastasis that came from different origins, for example. So we took single cell data of brain metastasis that came from melanoma, lung, breast, different origins, and we want to know what are the common molecular factors that explains why they become brain metastases. So what he did was to integrate all this single cell data. He identified the cell types, but what we wanted was actually to see the communication between the cells. We were specifically interested on how astrocytes communicate with endothelial cells, because there is already evidence that astrocytes plays a major role in the blood-brain barrier. So what we did was to look at this to see how the astrocyte was sending signals to the endothelial cells. And then we look at the differentiation parts, how these factors differentiate along this process. And we confirm this using spatial transcriptomic data to see if these factors, where they were specifically. And then we come up with a model that tries to explain how the tumors, the primary cells leave the tumor, goes to the bloodstream, past the blood-brain barriers, goes to the brain, and then gets established there. All of this using public available data. In another paper that we are doing this similar stuff, work is with pancreatic cancer, where we are also trying to identify specifically one type of cells that we think it's driving this aggressiveness of pancreatic cancer. And we are also using spatial transcriptomics to try to find what are the roles of some of these genes. And then we had to come up with novel spatial transcriptomic tools, but because this field is very, very recent, I'm going to mention the first tool that we developed, which is called Archipelago, but I'm not going to tell the story of Archipelago, just the application. What we do, for example, is to take, for example, an image like this one, which is a liver with a cyst, which is a parasite, and then we can use the Archipelago with the genes from the parasites to identify where the parasite is. And then we can do the same now with, for example, look at inflammation, where inflammation is happening, and then you see beautifully surrounding the cyst, and then you can compare how distant biological processes are, how distant autophagy is from type 1 interferon, and using this kinds of tool. Another tool that we develop is called Epispatial, and Epispatial starts from this biopsy. This is a lung cancer biopsy, and then you do the spatial transcriptomics where you identify the cell types, where is fibroblasts, where is macrophage, and then you can do an epidemiological approach using epidemiology to analyze this. And what do you mean by epidemiology? Well, in epidemiology, you try to identify why there are some regions where you have a lot of disease, and then in the middle, there is a region with no disease. What's going on there that explains why the burden of the disease is much lower than the surrounding, okay? And the opposite is this one. It's called high-low, where you have no disease, and in the middle you have a disease, okay? So what we thought is this is the same problem in a lung cancer, in a cancer biopsy. You want to understand why there is a place in the biopsy with a lot of cancer, and then in the middle there is an area with no cancer. What's going on there? What are the cell types that are there? What are the genes that are activated in this region that is supposed to have cancer? And then we did this to identify these regions called low-high, high-low regions. Then we can identify also the border of the tumor. So each image here is an image that becomes a dot in this box plot, where we can try to see what is the cell type that is consistently located in this low-high regions and which specific cell type is there. We can also use different technologies with single cell technology where we identify exactly the cell type, and then we know the subsets of the cell type. For example, we see that in this case, specific subsets of macrophage were specifically enriched in these low-high regions. We apply this to infection as well. This is a data set of spatial transcriptomics of the brain of the mouse infected with Japanese encephalitis virus. And what we did was to first identify in the brain where is the expression of viral genes. So we know now where probably virus is replicating. And then we use another approach to try to clean the mess, to identify the regions that are specifically infected. And then we use a spatial clustering to identify 4 clusters in this image here. And when we do this, now we can make these rings where we can see the distance between the center of the infection towards the border of the infection. And when we do this, we can see some genes very nicely increasing when you go to the edge of the clusters, and some genes that decrease when you go to the edge of the clusters. Now I'm going to be very fast. Cognitive computing, what we did was to take 45 million papers, ask the machine to read the papers, and create these knowledge graphs where you connect genes, drugs, and disease, and then a human can extract information from this. We apply this approach to do drug repositioning for psychiatric and neurological disorder. Basically, what we did was to extract from the literature information about genes and drugs related to these neurological disorders. So that's how we knew that Alzheimer was close to dementia, bipolar disorder was close to schizophrenia. And then we can look at the molecular signatures according how they are described in the literature, not experimentally. Of course, there is an experiment that put them in the paper, but what we are looking here is the papers. And then we can see what are the common mechanisms between this disease based on the literature. And then we can try to do drug repositioning by looking at drugs that are targeting one disease, but because of the genes that are shared with another disease, you can suggest that it can be repositioned to treat another disease. So this is an example of Alzheimer, but we did with all the disease. And these antibodies or drugs were, if you try to find it in PubMed, you're not going to see a paper that says that drug is good for Alzheimer, because we check manually. But if you go to clinicaltrials.gov or the internet, you're going to see that actually there are companies already testing these drugs against Alzheimer. The computer vision parts, we start with Levac, which happens because in 2018, Brazil, there was an outbreak of measles in Manaus, and we lost the certificates of eradication of the disease. And when we analyzed the 7,000 cases there, we realized that 95% of the people were not vaccinated or were vaccinated a long time ago. And that was a problem for the government, because the government want to know how many of these people, what is the vaccine coverage in that population. And the problem is that the government didn't know, because the information was in the vaccination cards at somebody's home. So you have to hire hundreds of thousands of people to digitalize millions of vaccination cards. So the government asked us to develop an app that can take a picture of the vaccination cards, detect the vaccines and the doses, and then digitalize this information much quicker. And then that's what we did. And that gave us expertise to work with computer vision. So we applied this to study parasites, and also to detect mpox by looking at the skin lesions of people. We also have another project with babies, because I realized that the problem of the distress of baby breathing, when the baby is tired of breathing, that is actually done by looking at the baby. So the physician has to look at the baby for a few minutes to decide if he's going to intubate or using surfactants. And then I thought maybe a video, if we film the baby, we can translate this breathing into a pattern that can help physicians to take the decision. So that's me breathing normally. And then I went upstairs, downstairs a few times, and that's me breathing. So that was like 1 week after the talk. And then my postdoc had a baby, so he filmed his own daughter. There's no ethics committee here. But if we failed because the size of the baby chest varies a lot, depends on if it's term, a term. And then what we're doing now is trying to come up with a laser approach where we can project in the chest to estimate the distance between the camera and the baby, and then that's gonna make the more accurate. And we are also using edge computing, which is basically the camera detects the face of the baby, anonymize the data before we receive the data. And that's the paper of Loic Dupré and Vinicius, and probably the reason of why I'm here. And I'm very happy to be part of this amazing work. I'm gonna talk the way I give this results because I want to go to the AI part. But basically, multiple sclerosis is a debilitating disease, and one of the reasons of why it's debilitating is because lymphocytes can go to the central nervous system. Tell me if I'm wrong, okay? But and it use integrins and this to infiltrate this central nervous system. And there is a very expensive treatment that explores the situations where you block this from happening using natalizumab. The problem is that it's a very expensive treatment and not everyone get the benefits of the treatment. So what Loic's group came up was with a system where you can test the person before he start the treatment by taking the bloods, putting it in vitro, and then adding the natalizumab to see if there are morphological changes in the cells, and that information is gonna tell you if the treatment is gonna be a success or not. So what they saw is that if you look at the morphology after treatment with the antibody in responders and non-responders, there are some changes. But of course, if you do unsupervised analysis, you can see that these morphological changes already separates responders and non-responders. And if you do a more supervised analysis, you can identify dozens or hundreds of features that could separate responders and non-responders. And that's where we enter. Because what we're trying to help was, can we use machine learning to tell which features actually is more important in predicting who's going to benefit for the treatment? And we use a tool that we developed called Feature Combinator that runs behind this random forest. And I want to explain how random forest works for you to see that it's not that difficult to understand. Random forest works. Imagine that you have, in this case, 21 responders and 19 non-responders, and then you take the levels, the values of feature A, and then you place each person according to this one dimension here, and then the algorithm identify a cutoff that separates most of the reds from most of the blue people here, and then you put here, and then you do the same with these sides of people, where you try to split this even more by using another feature, let's say the size of the cells or the granularity, and then you do the same thing with the other side with another feature to try to split even more. And then you keep doing this until you have a decision tree where you separate all the responders from the non-responders, okay? But the trick is that you do not create one decision tree based on the entire data set. What the algorithm does is to first get a random number of features and a random number of patients, and that creates a random data sets that you use to create this decision tree, okay? And then you repeat this 1,000 times. You have 1,000 random data sets, and that's gonna give you 1,000 decision trees. And what is the name of the place where you have 1,000 trees? Force. Okay, so random forest is very simple algorithms, and then you can count how many times the features appear in these 1,000 trees, because that's gonna give you the importance of the feature in predicting something. And that's what we did to, in their data sets where they have 2 independent cohorts. And what we run, the feature combinator, was to try to identify what is the minimum number of features that can predict well who's gonna benefits from the treatment or not. And I have plenty of other applications that I want to talk to you later. So generative AI, this is to show that we have a TCC, like I don't know how to say it in Portuguese, in 2019, trying to make these healthy robots here that before the pandemics, that's gonna help interacting with people. We have another project that transforms X-rays, which is a 2D image, into a CT, which is a 3D image. And we're using this tool called generative adversarial networks to try to make this happen. And this is an example of 3 CTs, real CTs of 3 different patients. And this is the CT made, the synthetic CT made from the X-rays of these people. We are also applying the same techniques to now remove a bias in our algorithms. That is, when we are trying to predict mpox, it works really well for Caucasian people. And because we have an underrepresentation of black skin, we can use AI to generate skins with darker tone, with lesions, and then train the AI on top of this. We have another project with disinformation and vaccine agency, and probably you guys face this problem in the WhatsApp group of your family during the pandemic. You receive a lot of fake news from your relatives, and then you have like to decide what you're gonna do with this, right? So if you're gonna try to answer all this fake news and get hold of this, or you're gonna leave the family group. Or in my case, the family group creates another parallel group so they can talk about this fake news without me. But because of that, in CNPq, they gave me money to create this multimodal WhatsApp bots that actually handle this for me. So it can receive text, audio, and image, and then it can tell if this is a fake news or not. So this is a real fake news. COVID vaccines cause mpox, but the image I created. And here is how the chatbot works. Of course, it works in Portuguese, but this is in English. You can say, can I get mpox if an infected monkey bites me? Which is true, and say, yes, you can, but that's very unlikely, but in theory, yes. If I put lemon on the lesion, can I cure? No, you cannot cure mpox with lemon. And then you put this image, and then it says, oh, that's not a true claim, this is fake news. And then you send an audio saying like, oh, my uncle said that he got mpox from the bathroom. And then it says, look, I understand that you're concerned about your uncles, but it's not likely that he got from there. You could, but so it works really well for the disease that we train. We train with dengue, mpox, COVID, and even the fake news that we did not train. The bots realized that there was a fake news. And one of the problems of why we did not give this back to the government is that nobody's going to use a bot that is going to make you feel stupid, right? That was the problem with the pandemics, because your uncle sent the fake news, and then you reply with 3 different New England Journal articles, and your Christmas is ruined. But what we are trying to do now is to try to make an empathetic AI, meaning that an AI that tries to understand why the person believes in this fake news and then try to convince with patience that we do not have as a human. So as a conclusion, just everything is changing. We start from this information economy where everything was gathering data, producing data, to the attention economy, because now we have a fixed amount of attention and endless distractions, to an AI economy, which is now AI telling you what you have to look at or study or do. Same thing with medicine. People start Googling the symptoms, and then after they start trying to find the influencer that's going to help him to solve his problem. And now AIs can call you and talk to you, and you don't even realize you are actually talking to an AI. And I don't know if you watch House, which is an amazing doctor. He always pick who has lupus or not. But if you look at him as a human person, he's horrible, right? He's rude. He doesn't like patients. He likes to solve the technical challenges. So these doctors, they are going to be replaced by doctors by AI. So AI is going to replace physicians that only cares about technical part, because that's the part that we can automatize. But the empathy, the connection, that's, I think, it's going to last longer. With that, I'd like to acknowledge that the supporters and also the team of my lab that did all this work that I'm showing you. That's it, guys. Thank you.
Yes, please. Renaud, you can come up there. Nice. Thanks a lot, Nakaya. And now I invite Renaud and Elder to be in front. And Zilton, you take my turn now, and Adriana is online. They can.
Adriana, you're there? We cannot listen to you. So I start.
Oi.
Oi.
Hello. Hi. Sorry
hello.
that I couldn't be there.
Can you see us?
A party emergency here. Preparing for our.
But can you see us?
I can see you guys. You're a little bit
wonderful.
far. But
Wonderful.
I know. Elder, I know.
Very good.
I would like to see your faces, but it's not possible,
me?
right?
Myself? You are not seeing me? Yes?
You guys. No, I see you very small on the edge of the screen.
Great.
Okay. So let me just check something here. But thank you very much for the two talks that were really amazing. And I think, well, I have some things to talk with Elder afterwards. But right now, I would like to talk about the immune checkpoints. Because, I mean, immune checkpoints, actually, I don't like the name, but anyway, it was established like that because they're not real checkpoints. They are regulatory molecules that come up after the cells are activated, usually. And one question that I was preparing to ask, but then you said it, is what about the role of these molecules interfering with the regular homeostasis, because that's what they are made for. And one example that I think is very, it's known, is that PD-L1 and PD-1 are necessary on the germinal center to get the B cells going, interacting with the follicular T cells. And PD-1 and PD-L1 is important on that, but there was no report in the literature for many, many years. Even on the PD-L1s and PD-1s knockout animals, we couldn't see, or they didn't really approach that. And then in lymphoma, they like to use PD-L1, or when they don't know what to do anymore, the physicians, they use the anti-PD-1 antibody. And my question was, it's more of a general question. I'm just giving an example with the PD-1 and PD-L1. And like on this case, what I used to imagine is that this interaction was really important not to blow up the lymph node. So the cells would copulate, and for helping proliferation is not necessary at all. So they would keep together, but the B cells activated have all the signals to keep the T cell going. So you need the signals that will shut down the some aspects of the T cell activation. So I'd like you to comment a little bit on that. And on the, I think the part I like the most is the modulatory role of these molecules, because like for PD-1 and PD-L1 on a terminal center, you need the cells to cooperate, to be active, but not to proliferate, otherwise you lose everything there. So I'd like you to comment on that a little bit, please.
Yeah, do you hear me?
Yeah, sure.
So I'm not a total expert about PD-1 in this context. What I know is more the example of BTLA and HVEM, which were shown to be very important in the germinal center, in the cooperation between TFH and B cell. And what has been shown in this case is that somehow this interaction between BTLA and HVEM, which is expressed by germinal center B cell, is very important to negatively regulate the co-stimulation provided by CD40 ligand from T cell to B cells. So somehow you get a signal that is important for the germinal center B cell maturation, but you need to control to some extent how co-stimulatory signals that are induced by the T cells would not be too strong so that germinal center B cell would be hyperproliferative. And there was some identification, I think, of patients that have lymphomagenes that have some mutation in this pathway between BTLA and HVEM, where this pathway is not, in interaction between the HVEM and BTLA is not efficient. And you see that this preventing this interaction of this inhibitory receptor is triggering this, potentially those cancer of germinal center B cells. So I think there's a way, this is a very nice way to, at the same time, make T cell being able to provide some co-stimulatory signal, but adjust the way those co-stimulatory signals are triggered to diminish them so that you don't generate too strong things that could lead to potential cancer and normal proliferation. So I'm not directly answering the question. I'm trying to find a way based on what I know on BTLA and the context you mentioned.
Yeah, thank you. That's exactly it. I think people, I mean, we've been using these immune checkpoints inhibitors without thinking on the consequences of that. Of course, if it's for a short term and the immune system is totally ready, probably when the antibody is cleared up, the person will be back to normal in a while. But in terms of consequences of that, like we see a lot of autoimmune responses that accompany the treatment or the treatment response, which is actually at one side is a good signal. It means that it's really working because these checkpoint molecules are important to prevent autoimmunity. Although, and we know, as you mentioned at the very beginning of your talk, we are a whole bag of autoimmune cells or autoreactive cells that do not cause disease, but they were positively selected anyway, anyhow. And so I think we need to think about that in terms of the function, the physiological functions of these molecules. And I was curious on when you show on your T-gate, I think knockout, that you have let's say, developmental arrest for CD4s and CD8, it stops, or it's accumulated on the double positive cells. Did you look at the repertoire
Yeah, we know.
of these guys to see if they're different?
Yeah, we
To
know
which
it's.
extent would this molecule be interfering on the selection process itself?
Yeah, so we know the repertoire is affected. What is very strange about this molecule BTLA is that, when you take it out, when you do a knockout, you don't see any major effect on T cell development. So you wonder, why would you have this molecule expressed at this stage where you have no main effect? And when you cross those knockout for BTLA in the Themis knockout mice, you see that it has an effect because it completely restored the development of T cell. And this is very specific to BTLA because I didn't put it, but when you do that with PD-1 knockout, you have absolutely no effect. So in fact, there's probably, so we have an effect on the T cell repertoire. And it's, my presumption is that there's a very subtle interplay between the affinity of the TCR for the self-antigen in the thymus and the expression level of BTLA and Themis. And the ratio between these molecules is going to be very different according to the strength of the TCR for self-peptide. And this is a way, I'm not going to get into the model we suggest, but to probably enlarge the TCR repertoire that emerged from the thymus and to enable the selection of T cells that would not be otherwise selected, either because the affinity of their TCR is too low, or maybe because it's too high. And it might be also a way to select T cells that have a higher TCR affinity, which is probably one of the goals of positive selection, is to enable the exit of T cells that have a relatively good ability to recognize MHC molecule. So I think it might be important in this particular regulation mechanism.
But do you think like on, sorry it was not clear, on migration or on TCR or T cell APC VDT?
I think it's mostly...
TCR strength of signal.
No, I don't know about migration, but for me it's more a question to reach the threshold or to adjust the threshold of positive selection so that T cells do not die either by neglect or either by negative selection. And by playing on this ratio between this inhibitor, BTLA, and this molecule that block this inhibitor, you can enlarge the repertoire of the TCR.
Did you look specifically on regulatory cells? Regulatory cells?
No, in fact we did not, but Themis is probably one of the most downregulated. Yeah. So it's a very, it's a protein that is highly variable in T cell subset, and it's extremely, when we do that.
And BTLA4?
BTLA is expressed on Treg, but so far there's no major function that was shown, and we haven't looked at it personally.
And it's from the beginning, like the thymic Tregs also have it already?
I think so, yeah. I think so, but I don't know whether there's a huge difference in terms of expression between BTLA in conventional T cell and Treg. I think it's more the level of Themis that is highly variable.
Okay, so thank you.
Thank you.
And let me see.
Bonomo, can I open to the audience also to make some question here?
Oh yeah, sure, of course.
So anyone wants to challenge?
I
have one. Hi, Adriana, Martin.
Hi, Martin.
But the question is not for you; it's for Renaud. So I think we discussed this last year actually, and it was beautiful again to see this overview and the contributions you brought at the very end. And I was talking a little bit with Elder about this during your talk, texting him. But let me put the question maybe in a different perspective, and I would like to to listen to your thoughts about it, because you have been discussing with Adriana whether this might have a role in the repertoire selection, and in the periphery, whether this might open a little bit wider the repertoire for the cells that would be in the range for getting really activated and expanded. The question would be more and not specific to one of the inhibitory receptors, which I think they're not really checkpoints. They're really, fine-tuning this. And I like your view on that because we agree on that. How would that be an attenuator for more specific situations linked to inflammatory responses or linked to insults to the cells, and not putting this only in the perspective of APCs or whatever? So how much do you think those, and maybe this might be one of the answers why we have so many of those inhibitory receptors, can be attenuating inflammation, such as we see for other. So I think the interferon PD-1 axis is very, it's a classic example of this, but how much do you think we might see of different circuits being inducers of the expression of these inhibitory receptors following damage, and infections, why not, but also other insults to the cells?
Yeah, so it's an interesting question. So what I try to think quickly about what you said about my personal knowledge on, for instance, BTLA, it's clear that those receptors, to what I think, are not just negative regulator of the TCR. They have a way more broader receptor that they target. And I know, for instance, that BTLA, which I'm studying more particularly, there seems to be a very strong negative regulator of cytokine receptors signaling like IL-12 or IL-7. So I think it has the propensity to at least, I don't know if it attenuates inflammation, but it attenuates the capacity of T cells to answer to respond to inflammatory cytokine. But I think it's not a field that is quietly known. What is also interesting is what I was trying to say about PD-1 is that this molecule, they seem to be able to work independently of the ligand that they recognize. Just by being expressed at the surface, they can connect to other receptors like the TCR. And I think, but other receptor and probably cytokine receptors. So I think it's also, and you have pathways that are very different, but also very similar between the cytokine receptor and receptors such as the TCR co-stimulator receptor. So I think they have the capacity to inhibit that. Now they're also expressed by cells that are very more pro-inflammatory than the regular T cell, like a antigen-presenting cell, myeloid cell, and all that. I think the role of those molecules at the surface of those cells is very unknown. That's what I was trying to say. And I would bet that it has the capacity to regulate how those cells are controlled. And I think there was some study from Vicky Buciotti, a student of Arlen Sharp in myeloid cell, that showed that part of the treatment of anti-PD-1 is by controlling the inflammatory capacity of the myeloid cells, or the way they develop into inflammatory cells in the tumor context. So there are knowledge that those receptors can also develop how the cells or become pro-inflammatory or not, acting on how they differentiate into those different type of cells.
And will this, mechanisms or this system, if we look at more broadly, apply also for non-immune cells?
So that is also super interesting because the cells, PD-1, for instance, PD-L1 are expressed in the CNS. So there's clearly a field of investigation there that is to be done. What of the very, and it relates to the question that Mariana asked before, is that those treatment with anti-PD-1 and all that, we presume that it's affecting the exhaustion and T cell, but those receptors are expressed very broadly in the immune system, but beyond. And you can expect that many of the effects are due to other cell type. And I think one of the probably promising approach, a potential approach, I don't know if it's promising, would be to more specific target those antagonist antibody to specific cell types that are maybe more appropriate to have a more selective effect rather than and avoid those immune-related adverse events.
Okay. So I would propose now a question from the students. So, and then we move forward. Okay. Anyone? that wants to ask something? Don't be shy. You can ask even in Portuguese. We translate. There's no problem if you are shy about this. Any? Milena? Because I know it's just because of that. I'm just joking. Anyone? Go ahead.
I don't know if I can give a spoiler. I don't know. Can I ask either of them?
I'm gonna move to Nakaya.
Yeah, that's the.
So
okay. To ask Nakaya, yes.
Okay, go ahead.
Okay, we were paying attention to your presentation. The part you talk, I think was. I think it was the case with arthritis, atopic arthritis. Yes. So the graphics you showed the, what was the name?
Diferença sexual.
Não, o nome da droga.
Inibidor de antagonista.
So Akira. Anakira. Yeah, Anakira. Okay. So that's a drug that inhibits the antagonist. Okay. So we actually in our course we are analyzing a case that has a mutation in that gene as well, and the curiosity is the sister responds to a treatment different than the brother. They both have the same mutation, and your presentation was talking about the difference in sex. And for us was like, oh my God. It's not really a question, it's just that I found it really interesting, the result, and for us it's very useful because we were trying to understand what was the difference, and that's the whole purpose of what we are doing in this course. So thank you very much. We're going to read the paper because definitely will be useful.
No, thanks for the question. I actually, that could be our first project then. Like, if you guys are having this family with this, I'm dying to start collaborating more closely with Zilton.
Okay.
That could be
No, let's then already move, to Nakaya now. Thank you, for everything. But, I just want to make a quick comment on the what Elder was showing to us before, and even before it started, just to think about in a more general way about the data that you were showing, and many ways of seeing, right, the data from different groups and trying to integrate that data. So you are really working with integration of different papers and layers of information and so on. So this is one of the tests that we are trying to do in our project, for example. But I would put, even one thing that you touch super quickly during your presentation, that is the different time of life, right? So if you have a pregnant woman, maybe you have a big change, in the life of the person. And then in that particular time, maybe all the transcriptomics, proteomics changes a lot, And we have another problem, when we started in our network to work with, because we work with kids. And we have few information about other omics in kids, in comparison to adults, for example. And this is also a challenge. So it's just to provoke, and to see from you, what are your thoughts, to better integrate not only the data that you are showing that you can integrate, but also if we can even make those cuts, in the life of the person, and to help to analyze the data that we're going to generate, but take into account also the phase of life and things like that,
That's a great question. I talk about sex as a covariate, but age is also a major covariate, and I completely agree with you. Like, we have a project during the COVID that I did not show, where we analyzed 10 million results of lab exams, from everything. And then what we noticed is that when you do a blood test, usually it says like, oh, your platelets is this. It's if you are 18 years old or higher, older, that is your reference for you, right?
It's the same.
Yeah,
It's
but when we
it's not-
notice is actually when we analyze millions of things, we see that over your age group, like there's a major change in the age that is actually, and this year it came out a paper that shows a individual baseline. So every person has your own baseline according to the, and I completely agree with you that we have to look at this more carefully. There is another paper with proteomics that shows that you can capture biological age much better than chronological age, and that I'm pretty sure influence a lot what you guys are seeing.
Good, thank you. So let's go. I know that Andrea has a question, and Adriana also. So we have many questions already.
Nice talk, guys. Elder, I was thinking about autoimmunity as a whole, because we know that there is a prevalence of autoimmunity diseases in female. Every disease you think, you see a different balance between male and female. And you also found some genes in neutrophils, which is a very common and basic cell in the innate response. Do you think that you have some basics for all autoimmunity disorders in those genes first? And thinking about aging, I was thinking about the impact of, getting in the sexual activity for females. This is a very change for a girl to be a woman. And do you think there is something tricky in that?
It's a great question. Yes, I think so. Neutrophils, immature neutrophils, I didn't show, but we have other studies on sexual dimorphism of disease from acute disease such as COVID and sepsis. We see major difference, and always immature neutrophils are the biggest discriminators. But I think this is more like a response than the cause. That it's my opinion. Maybe it's because the sexual hormone is driving more leakage or whatever, and that is causing this exacerbation of the autoimmunity. But the cause I didn't show here as well. But I was telling Vinny that we took thyroids, blood RNA. Bulk RNA-seq of thyroids of hundreds of men and hundreds of females that did not have autoimmune disease in thyroids. And then when we measured the expression levels, we saw that there were more genes related to antibody secreting B cells, suggesting that there is more infiltration of antibody secreting B cells in female thyroids that are normal. And then we look at single-cell data of thyroids, and we saw a lot of antibody-secreting B cells in the single-cell level, proving that, in fact, there is more infiltration or differentiation there in the thyroid. And then we look at spatial transcriptomics of thyroids of females, we saw it again. And then we found a paper in 1970-something that shows in microscope that there is a lot of, there is evidence of ectopic germinal centers in the thyroids of females. So what I think happens in some cases is that's probably driven by sexual hormones. There is a predisposition caused by this differentiation ectopically. That's the interesting part, like in the tissue that's it may be influencing. And I'm very interested in trying to understand this. And I was even going to talk to Loic to look at neutrophils from women and men at using this image system to see if morphologically they are different, because I bet that you can find differences.
Very good. Adriana, your question to Elder?
Oh, can you hear me?
Yes, perfect.
Okay. My question is, well, first was something related to that. Because we end up coming up with so many other questions of basic biology, right? So this is one use of the IA that is really great, that it generates answers, but also it generates tons of detailed or biological questions that could help on the therapeutics for the data you found. So is something really wonderful. So you need a lot of collaborations for wet batch, right? Now my question is about the fake X-ray, the fake tomography.
Yes.
How many X-ray images do you need to be able to do the fake tomography?
Well, we say synthetic tomography to not be like a fake. But I understand. I understood what you meant. But so the synthetic CTs we made with 3 independent cohorts of some dozens of CTs and X-rays, and now we are expanding to thousands because the problem was to find this data set where you had the pair of X-rays and the CT to train the AI.
No.
But you can actually get the CT. You can actually get. Oh, sorry.
How many? Because I don't know how you do the synthetic tomography study with an X-ray, but for sure. Well, what I thought was that you would need some different X-rays to
from the same
pile
person,
them
you mean?
up.
From the same person, you mean?
Yeah, from the same person.
Yeah.
Yes, I now understood. So there is a technique that if you take the picture of someone 2D, you can estimate how the face is going to be 3D because human faces don't change much. Of course, you don't know if the person has a ponytail, but you can definitely estimate how the ear, everything looks like in 3D by just looking at 2D.
Wow. Okay.
And the principle is the same by looking at the shadows and several patterns that you have on the 2D image that humans cannot tell, but you can estimate how it's 3D. We do have some data sets where we have the front and then the sideways X-rays to see if we are getting right. And we are definitely do not expecting to replace CT using this technique. What we want is actually to imagine like a physician in the middle of Brazil where the city don't have a CT. And then the physician has to decide if he's, based on the X-rays, if he's going to send the patient to the capital and spend a lot of money, or if he can get a little bit more information from that X-ray. So what we want is to actually increase a little bit of more information from these X-rays, but always giving the decision to the physician, not making any diagnostic or anything. So we are creating like a platform, a web tool that the person can upload the X-rays and then see the CT, and then decide if he's gonna actually send to the real CT, not the fake one.
Very good. Very good. So let's go to the student. There is one student here. Yeah. Let's see.
I'll come back later.
Okay.
Hello. About the neutrophils, you saw the difference between female and male, and you saw immature. And how about extracellular traps? Did you uncover extracellular traps, neutrophil extracellular traps? And why the female are more immature neutrophils? It's about the density of the cell. And I found really interesting the RAB8 genes about the vesiculation. Is it somehow related with the vesiculation of neutrophils itself and the system of endosomes?
That's great questions. So the neutrophils part, we have not explored nets or other mechanism of why they are different. It's very descriptive in a way that we just see that the levels change. But now when we look at these 44 genes that were related to neutrophils and females, several of them were also related to nets and some interesting process. But we're not claiming knowing the mechanism. But I do think that's very interesting. For the RAB8B, I did not show here, but we did the structural biology part, the docking. So we saw that RAB8B was important for the SARS-CoV-2 to go inside the cells. So we prove at least computationally how probably RAB8B is helping the virus to get into the cell. but that was computationally.
Good, thank you. Loic has a question here.
Hi, I like both your talk of Renaud and Nakaya, and actually my question is a question around bridging the two talks, or the dimensions from the two talks. So Renaud discussed about very complex mechanisms, you know, that are underlying those many receptors that are expressed, we understand, not only in T cells, but many cells in the organism. And he presented list of possible mechanistic regulation to explain the biology behind those receptors. You, Nakaya, from your side, you presented beautiful ways to exploit already generated knowledge, but that mostly comes from, I mean, not only, but mostly come from RNA sequencing analysis, so it's mostly looking at genes in terms of level of expression, and you showed beautiful ways to connect that with medical records, with disease evolution, prediction of treatment response, etc. So my question, trying to bridge those two dimensions, is how much AI would now be able to help building models, mechanistic models, you know, to go beyond just the expression level of genes, but how they really work together and how they mechanistically regulate cell function at the cellular level, but also at the organismal level. So yeah, I mean, there are different questions here. One is maybe a very simple one. How do you think AI is today equipped to maybe help someone like Renaud to decipher among the many ideas he has about how his system could mechanistically work? How can AI help him, assist him in maybe designing a model that would actually integrate those different mechanisms that he was telling us about? And the other question, the one is how to help Renaud, and the second question is how Renaud can help, you know, with his more mechanistic studies, help you enriching your AI prediction, for example, with more mechanistic information, because I believe understanding more in depth the mechanism will also allow us to define treatments and to find vulnerabilities in the mechanisms that can be druggables, right? So it's nice to know that this pathway is potentially relevant, but understanding mechanistically how things are working, we can have people here with, you know, protein structure knowledge that can really design, you know, the drug to target a very precise mechanism. So, yeah.
When I was watching Renaud's talk, I was having like 10 different projects to start like with this. And some are related to AI, some are not. One is the diversity of combinations that you show that this receptor in this, with this co-receptor is gonna trigger different stuff. So just organizing this information, it's very helpful for me to try to model with AI something more mechanistic, you know, like putting this in a formal way. And one of the things that we are doing that I didn't show, that's if you have a network of A connecting to B that triggers C, and you can only fill part of the network with information, expression or levels, you can use AI to actually estimate what's going to happen with the rest. You know, it's a very complex stuff that not even me understand, but we would love to test this with you, meaning that you have underneath complex network, you put what is already known, which is partially resolving this network, and then use AI to try to have the full picture, and then you try to predict if this is true or not. So that can be doable now because, or tested at least now. The other thing I think AI can help is with the structural parts. I don't know if you know that there is a ChatGPT for proteins, meaning that instead of tokens being words, predicting the next words, you can actually train transformer with amino acids being the tokens, meaning that you can actually design a a co-receptor that's going to work the way that you think, almost like by talking to this. It's like synthetic biology, but applied to this immune checkpoint would be like awesome. And I'm pretty sure someone is already doing this. But what I would love to try to help or somehow contribute to with this. Yeah, and that's
so we are running out of time, right? Okay. So we are very happy to have your both talks here. It was wonderful time. I think everyone here are very happy about. Can we give an applause to them again? Obviously, again, happy birthday to Elder. It was great to have you here today. And okay, we continue, right? So the course is not over. It's only 1:30. Only this session is over, okay?
Tchau, Adriana, também. Muito obrigado.
Tchau, gente. Desculpa, daqui a pouco eu tô aí.
Esse é